Hi, potential Beckman scholars. My name is Christine Picard. I'm an associate professor in the Department of Biology, and I'm the director of the Forensic Sciences program here. And I'm going to tell you a little bit about my research. Most of the work that I do, or that we do in our lab, is around insects and trying to understand the correlations between insect genotypes and their phenotypes. And specifically, we're interested in some development rate variation, both for forensic applications as well as a sustainable alternative protein source application. So my work spans these two different areas. And we use a combination of bioinformatics and genomics, as well as standard molecular biology tools. So rather than me telling you about what our lab does, why don't I introduce you to the people in our lab so that you can actually see what we do. So let's go into the lab. Let's see what we've got going on here. Hey!
Hi.
What are you up to?
I'm taking pictures of flies so we can use an AI system to be able to figure out which ones they are.
Yeah, and you are an undergraduate
Yes.
in
Forensic science.
Awesome. Okay, let's see who else. What do we got going on over here?
Hello, I'm Hector. I just started here at the graduate biology program. I'm studying the black soldier fly genetics. Right now I'm just looking at one gene, yellow. It's named for the color that they turn if you don't have it.
And what is the purpose of the work that you're doing?
Well, I am studying the black soldier fly because we are using it in industry, and there are certain traits that are useful to have, and we're trying to see what's the difference between those that are domesticated and those that are not.
Awesome, yes. Black soldier fly is the hot new insect for a sustainable alternative protein source. Let's see who else we've got here. Oh, who's this?
Hello, I'm Frank Lawrence. I am a PhD student here in Christine Picard's lab. Currently working on some genomic DNA isolations from C. rufifossés. We're currently studying the genetics of this fly because it has two females, one that only has female offspring and one that only has male offspring. We would like to know what the genetics are behind this mechanism.
Awesome. We have some really cool flies in this lab if you're interested in this. What do we have over here? May we interrupt you for a minute?
Sure. Hi, I'm Ashton. I'm a second-year master's student in the Forensic Science program. And my project deals with blowflies, which we know feed on feces and carrion. And by analyzing the DNA in their gut contents, we can determine what animals they've been feeding on, which tells us what animals are currently living, as well as what animals are deceased in the environment.
Awesome. And what are you setting up here?
I'm just about to set up some PCR.
Excellent. Thank you. So our lab has standard molecular tools, as well as you saw with Hector, bioinformatic tools. So we have a lot of computers in the lab where people are working. We have one final stop to go to, but we have to go up a floor, so I'll be right back. Okay, well, we've moved upstairs to the third floor, and this is where we keep our fly room. So let's see what's going on in our fly room.
Hello, I'm Khadija. I am studying the effect of relative humidity on blowfly oviposition.
And then what are you actually doing right now?
I am just changing out the water bottles. You know, flies get thirsty too.
And she is really good at making sure those flies stay inside those cages. And let's go see what's happening in the other room.
Hello again. I'm just looking at my larvae.
And what are those exactly?
These are Lucilia sericata. They are a blowfly native to Indiana.
So this is our what we like to call our maggot room, but this is where we have all of our incubators, so we can do controlled experiments with our maggots to be able to understand various biological aspects of them. So that concludes our visit in the Picard lab. I do hope you'll consider coming to do some research with us. We love having students in the lab, and you'll get to learn quite a variety of different aspects. So feel free to reach out if you have any other questions.